left my clicker up in the lab, but I just have to keep walking over to the computer. We started the last, again, it's chapter 12, not chapter 10. Last class, we started talking about all the different ways we can stop bacteria or microbes from growing out in the environment, so out on surfaces. This chapter goes hand in hand. It's also things that are meant to control or kill bacteria or other organisms, but in the body, because what you would use on surfaces are generally completely different things that what you would use in the body. On surfaces, we can spray things, we can incinerate things, we can do lots of different things out on surfaces to kill microbes, but we can't do those same things to kill and inhibit microbes in the body. So, this chapter, we're just going to go over some terminology when we're talking about different antimicrobials. What are the mechanisms of action, which really is how do these different antimicrobials work? How do they kill? What are some considerations and which one we're going to choose to prescribe to a patient? And then now the overuse of antimicrobials is leading to the resistance of a whole bunch of these different types of drugs. So just some terms. So the word drug itself is used all the time, and the word drug just means a chemical that changes the body's physiology, which is like a really boring definition. But it's a drug that changes the body's physiology. So it's a pretty generic term. And we talk about, we're prescribed all these different types of drugs when we go to the doctor. Well, there are lots of drugs. I mean, there's not just drugs that you get prescribed at the doctor. There's illegal drugs. And, these are just a generic term for any kind of chemical that's going to change the body's physiology. In this class, we're not going to talk about the illegal drugs, although we can talk about that when we get into the fungus. But we're talking more about the different types of drugs that are going to be prescribed to patients. So then we kind of narrow that definition of drugs down to chemotherapeutic agents. It's not fully narrowed down, but chemotherapeutic agents are drugs that are used to treat a disease. Drugs to treat disease. There are lots of different types of diseases out there that we don't focus on in this class. We don't focus on, multiple sclerosis. We don't focus on different types of cancers. But those all need drugs that are used to treat different diseases. We're narrowing it down even further into mostly what we're talking about this class, and we're specifically going to be looking at antimicrobial agents. So these are drugs that are used to treat, microbes that cause disease. So that's why they're called antimicrobial. So these are meant to specifically treat microorganisms or kill microorganisms. Whether it's a bacterial that's a microorganism, whether it's a fungus, whether it's a virus, whatever it is, these are drugs that are meant to treat a disease, but specifically diseases caused by a microorganism. So we call them antimicrobial agents. So anything we talk about in this PowerPoint, they're all antimicrobial agents. Now, there's lots of types of antimicrobial agents out there, and a couple different ways we group them is based on where we got them from or how we've manipulated them. The word antibiotic, the term means against something living. But antibiotics, these are naturally formed antimicrobial agents. I'll give you an example. So an antibiotic, these are naturally formed, naturally found antimicrobial agents. These are things we can find out in nature, and they inhibit microbes from growing. So they're antibiotics. We use them a little bit in different terms. We use the word antibiotic, and we kind of encompass it with everything. An example of a naturally occurring antimicrobial agent is penicillin. That inhibits bacteria. We've figured this out, that there are different fungus that can inhibit different bacteria, and this is where we actually got our drug penicillin from. It came from a fungus, which is a naturally occurring fungus, and we know it inhibits bacteria. So So we know there are lots of different types of fungus, there's different types of plants, there's lots of things out there that inhibit naturally different types of microorganisms from growing. Honey is actually an antibiotic. You didn't know that. Honey has a bunch of antimicrobial properties. Naturally found substance, it's an antibiotic. Now, a semi-synthetic is an antibiotic that we've chemically changed in a lab. A lot of times we just call it antibiotics. It's just easier to say. But a semi-synthetic is an antibiotic we've chemically changed in a lab. It means we've chemically changed it maybe to make it a little more shelf stable, maybe to make it a little more effective at how it kills, but we've chemically modified it a little bit. Taken the Penicillium fungus and we have modified it. It is where we get some of our synthetics. So people that are allergic to penicillin, is any of you guys allergic to penicillin? It's not uncommon to be allergic to penicillin. A lot of people that are allergic to penicillin are allergic to its semi-synthetic derivatives. Amoxicillin is actually penicillin that we've chemically modified and we've changed a little bit. So it works a little bit different, but it came from that Penicillium fungus. And because they're related, a lot of times if you're allergic to one, you're allergic to others. Not always, though. Just based on its name, is a completely man-made drug. It's not like we took something out of nature and just modified it. This is a completely man-made drug. And this is kind of where we're going to these days. There's not a lot of things out there in nature that we haven't discovered yet, but you know, we kill rainforests, we kill options, that we're going to these lab-made antimicrobial drugs. Now, when we start talking about mechanisms of action, this really is, you know, how do they kill? But for all of our different antimicrobials, again, antimicrobial just means against a microbe, there are different types of microbes that are out there. And so we need and want our different antimicrobials to have selective toxicity, which means toxic against certain things. So it's selective in what it's toxic to. We have drugs that are specific for bacteria. We call them antibacterial drugs. We have drugs that are specific for eukaryotic infections. These are going to be larger cells, usually parasites that we'll talk about later. We have drugs that are specific for viruses, antivirals. So they're selectively toxic against different things. And then even in amongst antibacterials, we have some that are selectively toxic. We're just gram-positive, we're just gram-negative, and they get narrowed down. But when we're prescribing drugs, we have to know what is it targeting. We want it to target what we want it to target and then not harm anything else. Now, the mechanism of action on how does it kill bacteria, how does it kill eukaryotes, how does it kill different types of viruses is all mechanism of action. Because you'll see me ask in previous things, well, what's this drug's mechanism of action? Which is really is asking, how does it kill? If it kills a bacteria, how does it do that? Because there are lots of different ways you can kill bacteria. You know, if it's going to kill a virus, how does it kill a virus? Or how does it stop the virus from reproducing? Because there's lots of different ways. So that's all mechanisms of action. And so this is a diagram. This is showing mechanisms of actions for different type of antibacterial drugs. So each grouping with each little picture is a different way that drugs can kill. And then it also lists under each different one some of the more common drugs that fit into those categories. So there are a whole group of drugs that inhibit the cell wall of a bacteria, that peptidoglycan. There's a whole group of antibiotics that inhibit the making of proteins. There's a whole group of antimicrobials that inhibit that cell membrane. Some inhibit metabolic pathways. You can't make the things it needs. Some inhibit DNA or RNA synthesis. Some inhibit the attachment. If certain things can't attach to cells, they can't get in, can't find a home, and they can't reproduce. So these are different mechanisms of actions. And yes, it lists some of the top common drugs. Out of the 6 different mechanism of actions, which mechanism of action do you actually recognize the most names of drugs? Cell wall. I'm going to say you might recognize a couple names. You're like, oh, I've heard of tetracycline and the protein synthesis, and maybe you've heard of a couple other, but these are the more common antimicrobial drugs that you've either taken yourself somewhere in your, lifetime, or people that have had these or been prescribed these, because really inhibiting the cell wall is the most common mechanism of action that we want to target. So, inhibiting the bacterial cell wall, most common antibacterial group of drugs do this. You probably recognize some of the names because it's super common. You've got a bacterial infection. Easiest way, sometimes the simplest way, is to stop the cell wall. The names, you probably, and if I say them wrong, you're gonna be like, wow, she's saying that wrong. It's one of those. How many of you guys are going into nursing? Are you guys all going into nursing? No, are you? Okay, because I was gonna say, I'm like, and I feel like Jasmine might be like respiratory. I don't think you're going to have to take pharmacology, but the rest of you have to take pharmacology. And it's one of those, I'm like, I want to take pharmacology. Don't be surprised if you see me in your class. Just because it intrigues me as well, and the spelling and pronunciation of everything. But this whole group of drugs, because bacteria have a cell wall, and it's made of peptidoglycan, I mean, it's initial armor. I mean, a building can't sustain if it doesn't have walls. Well, neither can a bacteria. Do our cells have cell walls? No, which means that's great, because we can take a drug that inhibits cell walls, which means it should have no effect on our cells, because our cells don't have cell walls. And so it's also a very commonly prescribed drug because we know there's very little side effects, just because our cells don't have any cell walls whatsoever, so it's not going to target or do damage to our cells. Now, what they prevent is they prevent proper peptidoglycan formation. That's what bacteria cell walls are made up of. I know it'll show a picture coming up, but peptidoglycan was made up of repeating NAGs with NAMs. Drugs, beta-lactams. So like, oh, we prescribed you a beta-lactam drug. It's a drug that inhibits cell walls. So what happening to the bacteria, like the picture, better than talking about it. So here's the normal cell wall. All these pinks and reds are your NAGs and NAMs. Again, it's the bricks that build the wall. So they're attached to each other side by side. They're attached to each other top down. I mean, they are a nice, strong, attached, solid cell wall. What these beta-lactam drugs that target the cell wall is they stop these things from attaching to each other. It's almost like if you built a building, but you didn't put any mortar between any of the bricks whatsoever. Not a very strong brick. And so what ends up, or very strong building. So what ends up happening, well, if a little bit too much water gets in, the cell's going to burst open because that cell wall is not solid. It can't withstand any type of water change whatsoever. And so it doesn't have those different attachments anymore. So it's a very weak cell wall. So that's how they work, is they prevent those NAGs and NAMs from attaching. It's just like building a building. Bricks don't have mortar in between it. Very weak cell wall, easily burst. Now, so what happens to the bacteria? The cell walls burst. And your cell wall bursts, you die. However, there are some bacteria, and we'll get to them when we start talking about specific bacteria. There are some bacteria that make an enzyme, that make an enzyme called beta-lactamase. So some bacteria make an enzyme that can break down this antibiotic, which means any of those antibiotics, the penicillins, the amoxicillins, are not going to be able to do anything because the bacteria is like, oh, you're going to take that drug? Well, we have an enzyme that can just break it down. So some bacteria are getting a little smarter against some of these different classes of drugs. It's kind of like, oh, well, if you give me this type of drug, I'm going to make an enzyme that breaks it down. These are some of the examples of the different drugs that are in this class that target that cell wall. So some of the semi-synthetic derivatives pretty much they're derivatives of the penicillin. The methicillin, amoxicillin, ampicillin, these really were all penicillins that we genetically modified in the lab. Other types that still inhibit the cell wall, and they either target different organisms different or work in slightly different ways about targeting the cell wall. So the vancomycin and the cycloserine, and vancomycin is a pretty common antibiotic given. These specifically target gram-positive cell walls. They're really looking at those thick cell walls of gram-positive. So people that have gram-positive bacterial infections, very commonly given that vancomycin. Bacitracin, again, it's still inhibiting cell walls. It's still inhibiting the NAMs and NAGs, but it works a slightly different way. So bacitracin, all those other drugs, are just preventing those attachments so that it's a weak cell wall. Vancomycin still affects the NAMs and NAGs, but it stops NAGs and NAMs from just attaching at all. Usually this happens in cell wall formation. So when the new bacteria are growing and they're trying to make more cell wall as they're growing and dividing, it prevents the NABs and NAGs from ever getting to the cell wall. So bacitracin just prevents the cell wall from ever getting made when the bacteria are reproducing. So it's not stopping the formations, it's just stopping the making of cell wall altogether. So it's still effective, it's still targeting cell wall, it just does it slightly different. It means the bacteria can't make a new cell wall. I'm gonna say these wrong, I'm like isoniazid and ethambutol. These are gonna target that mycolic acid in those mycobacterium. So that is that lipid substance that's found in the cell wall. And these are the drugs that are generally given to tuberculosis or anything else that would have that mycolic acid in it. They're still targeting cell wall, but slightly different. Another mechanism of action is in inhibiting protein synthesis. And so all proteins are made by the ribosomes. And so we're generally going to be targeting ribosomes. And we're going to be targeting that whole process of making proteins, the transcription, translation that's coming from the ribosomes. Now, whether you're a prokaryotic cell, like a bacteria, whether you're a eukaryotic cell, like any of our parasites that we're going to talk about, they all have ribosomes. They're slightly different sizes, but they still have ribosomes. A eukaryotic ribosome is slightly bigger than a prokaryotic. Does the exact same thing. Now, what it's specifically targeting when it's targeting the ribosome, it's targeting the translation process. There's transcription and translation. That's our next unit. Where we can take DNA and make messenger RNA and read it and make amino acids. It's specifically targeting that making the amino acids or the proteins that happens in the translation. Some of our eukaryotic organisms have mitochondria that even have ribosomes as well. Is that it can target the ribosomes and it can target that translation step. So one, it can sit there and target, and it can cause incorrect amino acids to get brought in. If you bring in a wrong amino acids, you're not going to make the correct protein, which means the cell can't do whatever it needs to do. Some, I'm not going to ask you all of these specific ways, but just know that there's a lot of different ways you can target protein synthesis and that translation. Some, you can bring things in that are going to block the messenger RNA or cause a blockage site at the messenger RNA. The transfer RNAs can't bring in amino acids whatsoever, which means you're just going to stop the making of amino acids on proteins. Some of these different drugs stop amino acids from attaching to each other. And so you can't make a long chain of amino acids if they can't attach to each other. Some are going to sit there and stop the messenger RNA from being able to go through the ribosome because it gets pulled through the ribosome and is read 3 letters at a time. Well, sometimes these drugs prevent that RNA from going through. can't pull it through, can't read it, can't make amino acids. Some drugs prevent that ribosome from even being able to attach and read the messenger RNA. Can't read it, can't make stuff. And then some are going to almost cause like a little tiny random mutation in the messenger RNA that part of the ribosome can't attach. So there's lots of different ways, lots of different types of drugs that all are going to stop the protein from getting formed. Again, just like all of our cells are making proteins, all of bacterial cells are making proteins, eukaryotic cells are making proteins. These proteins could be enzymes, these proteins can be structural material, and if the cells can't make it, they will not survive. Another mechanism of action. Hey, I messed up. I thought I had another slide. I thought I had more examples. Another mechanism of action is, we did cell walls, we did protein synthesis. This is disrupting the cell membrane, that phospholipid bilayer, that whether you're a bacteria cell, whether you're a eukaryotic cell, you still all have a phospholipid bilayer. And the easiest way to disrupt it is cause holes or pores in the cell membrane that shouldn't be there. Pretty much like poking holes in the cell membrane. It means things are going to get in that shouldn't get in, things are going to get out that shouldn't get out, and in the end is the cell will die. One of the most common drugs that's given that targets specifically the cell membrane is a drug called amphotericin B. It is an antifungal drug, so it's not targeting bacteria, it's targeting fungus. And it attaches to a structure called ergosterol in the cell membrane. So it targets something called ergosterol. It's like this little purple insert on the picture. And the drug recognizes ergosterol, and anywhere there's ergosterol, it inserts this pore. But we do run into a problem, because humans don't have ergosterol. So don't have to worry about that. But we do have something else in our cell membrane that's pretty similar to ergosterol in its structure, and it even sounds a little bit like the name ergosterol. Any guess? Something that's in our cell membrane. Sounds like ergosterol. Everyone wants a low level. Cholesterol, yeah. I was gonna say, I don't know another hint to say it. We have cholesterol in our cell membranes, and it unfortunately slightly resembles ergosterol, which means that sometimes if you have a fungal disease and you take amphotericin B, and amphotericin's great at killing fungus because of it recognizing the ergosterol and putting these channels in, but it can sometimes insert channels anytime it recognizes cholesterol in our cells, which means it can cause bad side effects. So it's super effective, but it can cause bad side effects just because it targets our cells as well. So humans do have ergosterol. Bacteria don't have anything that looks like ergosterol. They don't have anything that looks like cholesterol, so it's not going to do anything for bacteria. It really is strictly an antifungal medication. When we talk about fungus, which I think is the last PowerPoint of the semester, or second to last, you'll see amphotericin B come up a lot as, this is the top antifungal that's always prescribed. That is possible that can be given that still targets the cell membranes, that's against bacteria, and it's a drug called polymyxin. And it specifically targets the outer cell membrane of bacteria, gram-negative bacteria. Because if you remember, gram-negative bacteria have two cell membranes and a thin cell wall right in between. And polymyxin can target the outer cell membrane. It cannot get in past the cell wall to target the inner cell membrane, and it can't get past the gram-positive cell wall either to target that inner cell membrane. So it's specific for just gram-negative. It's not commonly given because that can also still cause side effects in humans. So it's kind of a last resort antibacterial drug that's given. Another way that these different drugs can work is they can stop metabolic pathways. So, if I have got a picture, I know I do. So, metabolic pathway is if we have substance A, it needs some enzyme, and then it can make a substance B. And then substance B can take another enzyme, and it can make substance C. And then it can use another enzyme, and it can make substance D. That's a metabolic pathway. I know in one of my previous pictures, it was like, well, a red triangle goes into an enzyme, and it shoots out little, green squares, or I can't remember the colors and the shapes. And I'm like, and a little green square goes into another thing, and it shoots out little yellow triangles. And it's a whole pathway to eventually get that final product. Well, we know there's lots of metabolic pathways that different microbes use, and that gives us lots of different options. If we know there's an enzyme that's needed for whatever substance A is to become substance B, we can target that enzyme, and we can just target that enzyme. And if the bacteria can no longer take A and make B, it can't make C, it can't make D, it can't make whatever product it needs, and ultimately it will die. If we can stop it somewhere along this pathway. And if we're like, well, we don't have a drug that works very effectively against that enzyme, well, we could stop it there. It doesn't matter. As long as we stop somewhere along that pathway, we target any of the enzymes that are needed to get to that final end result, we can stop that process and stop the bacteria from growing, stop the fungus from growing, viruses that we have, but we do have quite a few different types of metabolic pathway drugs. A couple examples. I thought I had a picture on here. A couple different examples. One drug called, I'm going to say it wrong, adaviquone. This specifically interferes with electron transport and that electron transport chain that we went over. So it's stopping that whole process of cellular respiration. That was a long process of breaking things down and tons of enzymes, and eventually all those electrons have to get somewhere. Well, this drug specifically targets the electrons moving. Heavy metals are really good for antifungals. And these are gonna, any type of heavy metals. Copper is a big one, which is why sometimes you hear copper bracelets and all that other stuff. Heavy metals do inhibit enzymes from working. Pretty much any kind of enzyme anywhere. Problem is, the thing can have bad side effects if it interferes with different types of enzymes we need. We do have drugs that block the activation of a virus. When a virus gets inside of a cell, there are certain steps that have to activate it. We have drugs that can target somewhere along those steps, which I think is one of my next slides. Well, it's the whole process of this needed an enzyme to become that needed an enzyme to become that, and so on. This is my ABCD. I just didn't hear that you knew any of these names. It was just an example. And eventually the end product is to make more DNA or RNA. We know we've got drugs that can sit there and stop the making of different types of these enzymes, which means we can no longer make those intermediate steps, and the bacteria or the fungus or anything else can no longer reproduce. It looks like a complicated picture. That's why I was like, I don't care that you know any of those intermediate steps. Just know it's a whole process, and we have different things that can stop anywhere along the process. We do have a couple that, again, are targeting mostly the activation of viruses. So it's the antiviral drugs, amantadine, and some other organic bases can target specifically the activation of viruses. And we have another that targets a very specific virus, HIV. And when we get to viruses, we'll talk more about HIV. But we know there is a very specific enzyme called protease that's needed to activate HIV. Well, we have drugs that can prevent that activation of that enzyme, which means HIV can no longer be activated and go out and reproduce and affect more cells. We've put a lot of money into HIV research, and we've gotten a lot out of it. So we have a lot of drugs that are used to treat HIV these days, and even prevent the spreading. other mechanism of action, I'm trying to think, this is my last one, is to stop the making of DNA. So we can inhibit the nucleic acid synthesis. That's your NA of the DNA or RNA. So it's blocking the making of DNA or RNA in transcription. You can't make copies of itself anymore because any kind of eukaryotic cell or prokaryotic cell has DNA or RNA. We can use these for them. Right now, we don't have a lot of drugs on the market that are specifically stopping the making of DNA and RNA. So most of the drugs that fall into this category are right now used more in the research level as they're trying to figure out how different things work. And we're using this a little bit as well to slow cancer cell reproduction down. So if we can target the cancer cells from reproducing, we're really slowing cancer down as well. The only place that I've seen that these drugs are actually prescribed or used is they are targeting the making of DNA by, I want to say, bringing in fake nucleic acids. So nucleotides, these are your A's and your T's and your C's and your G's. Those are your nucleotides. But we have drugs that look like A's, T's, C's, and G's, but they're not the A's, T's, C's, and G's. And the only one that's given that I know of right now is for HIV. So these are examples of nucleosides. So these are like fake nucleotides. So it's like your A's, T's, C's, and G's, but they are structurally similar, but not exactly the same. And if you take a drug that has these fake nucleotides, if you had an A T is supposed to get brought in, but it's like this little, fake T, which means it's fake, structurally different DNA, which means it's not going to be able to do whatever it's supposed to do. Again, the only time I've seen this used is in some of the HIV preventatives, the PrEP drugs. Prevention. But the idea is that when HIV is replicating, it is bringing in incorrect nucleotides, so it no longer is effective HIV. So it's not really killing it, but it's stopping the reproduction of the HIV virus in the body. The other ones that are out there, again, not commonly used. Quinolones, fluoroquinolones work against some of the bacteria DNA. It's an enzyme specifically. Some can inhibit RNA, and then HIV. Other than those fake nucleotides, there is an enzyme that is used in the making of the making of the DNA of HIV. It's called reverse transcriptase. Reverse transcription, and we can stop it. It's taking RNA and making DNA from it. Super unique and very specific to HIV, used just for HIV. So very few drugs on the market that are used that target nucleic acids. The only ones that I know of are specifically targeting HIV. Is preventing the attachment. So if we can stop different things from attaching, one, if it's a bacteria before it can reproduce, first has to find a home. If we can prevent that, awesome. Viruses, they have to get attached to our cells, get inside of our cells, and then hijack them. Well, if we can prevent them from attaching, we can prevent them from getting inside of our cells and causing infection. And this is where a lot of our new antimicrobial drug development is because this is, our newest area for preventing viral infections. Because viruses, we know, have to attach. We know we're looking at what specifically do they need to attach, where do they attach, and can we stop the attachment? If we can stop all viruses. Like we can stop the flu completely. We don't need this, annoying flu vaccine every year that changes. But we can stop HIV, period. We can stop any virus from ever causing disease. As long as we can stop it from attaching. If you can't attach to a cell, it can't get in, it can't cause a viral infection. Considerations on what drugs should we choose when we have a patient that is sick. One, We want a drug that's readily available. We want to be able to prescribe it, and they should be able to drive over to Walgreens and it's right there and ready to go. We want it cheap because no one wants to pay for a super expensive drug, especially the insurance companies. We want it chemically stable, which means it usually can be on the shelf. Some have to get refrigerated, but that becomes a hassle, and people don't like that. We want it easily administered, usually oral. We want it non-toxic, non-allergenic. No one wants to get sick from their drugs. We want it toxic against a lot of different things, not one specific virus or one specific bacteria. And we want to make sure it's effective against the patient's specific bacteria. And so this is where, identifying that bacteria becomes important, or other microorganism. So depending on how many things a drug is effective against, we group drugs into narrow spectrum drugs and broad spectrum drugs. So narrow spectrum drugs work against a very small group of organisms. Sometimes only one. HIV drugs are very specific and they only target HIV. That's about as narrow spectrum as you can get. So narrow spectrum, they're effective against a very small group of organisms. Broad spectrum, these are effective against a large number of microorganisms. Some examples I have on here that shows, what are they all effective against. A narrow spectrum would be the polymyxin that targets that outer cell membrane of just gram-negative bacteria, and not even all gram-negative bacteria. That would be a very narrow spectrum drug. It's targeting just a very small group of just gram-negative bacteria. Whereas something like erythromycin or tetracycline, it's effective against different types of bacteria. Gram-positive bacteria, gram-negative bacteria, and then we've got these oddballs, chlamydia and rickettsies, that they behave and look different. But they're effective against those too. That's a broad spectrum drug. And a lot of times, these are what are prescribed. When you go to the doctor and they're like, oh, you have a sinus infection, you have an ear infection. Did they take a sample, take it to the lab, and identify it? Do you walk out of there with a, antibiotic prescription? Know what it is. They just know you have an infection. What they're generally going to prescribe is one of these broad spectrum drugs that we're like, it'll kill whatever's in there. We don't know what it is, but it'll kill it. As we prescribe a lot of broad spectrum drugs. The only time we really start to identify the bacteria, especially with sinus infections, ear infections that are so common, is if you take these drugs and they're no longer working. Then it's like, we got one of these rarer things. Then we might actually need to identify it, narrow it down to make sure we give you the right bacteria. And then we also want to know, well, how much antibiotic are we given? Is it like a 10 milligram dose, a 100 milligram dose? We want to make sure we give enough of that antimicrobial drug that it's actually effective against your infection. And so there's different ways we can test how effective, or how, different concentrations and how effective they are. And one of these we're actually going to do in our lab in a few weeks. It looks like this plate. Called the Kirby-Bauer test. Another one's called an E-test. Is they want to know how much antibiotic can be given that we know is going to kill the bacteria, but we don't want to give more than. We don't want to give less than is necessary because then it's ineffective. One common way to know exactly how much we can give, it's called a little E-test, is this strip is dipped in different levels of concentrations of antibiotic. 256, maybe it's milligrams, but a certain level, and then it has different levels, decreasing concentrations. What we can do is we can put bacteria all over a plate, lay this right down on there, and we can look to see what is the minimum amount of that drug that we can still see it's inhibiting bacteria. If we give something this low, we know it's not going to do anything. The bacteria grow right up to it. We give this big, we just don't need to give this much. Why give 256, times the amount when we give 0.75? And so we can use this to know how much of that drug is effective against the patient's bacteria. Not always done. Again, it's broad spectrum, super cheap and easy. But sometimes we do have to do this if people are having hard to treat bacteria. These are just showing how we can do some of those minimal inhibitory concentration tests. What is the minimum of amount is that we can sit there and dilute out the different levels of antibiotic, put it with some bacteria, and we're going to look to see what's the lowest level of antibiotic that we can be given that we still see bacteria growing. I mean, that's going to be the lowest amount we don't want to give. We want to give higher than that. Then how is that drug going to be given? Is it going to be given as a topical? Because some people do have bacteria infections on the skin. Is it going to be some type of antimicrobial cream? Is it going to be taken orally? Is it going to be an intramuscular administration? Or is it going to be intravenous? So different levels or different ways we can give these different antibacterials. And this is to show the difference between oral intramuscular shot or continuous intravenous right into the vein, and the levels of concentrations of that antimicrobial in the blood. Now, out of the three, oral, intramuscular, and intravenous, which one has the lowest level of antibiotics that makes it into the bloodstream? Oral. Oral antibiotics have the lowest level of that antibiotic actually making it into the bloodstream. How are most antimicrobials given? Oral. But it's the easiest. And so people want easy. I want to be able to leave Walgreens with my drug, and I can go home, and I know I need to take my drug twice a day or 3 times a day. To get intramuscular, I would rather take something orally than give myself 3 shots a day. Or continuous intravenous, you're in the hospital at that point. You're hooked up to an IV. And I'm like, so what's the least effective for getting that drug into the bloodstream, concentration-wise? It's the easiest, so that's the one we give most often. Generally, it's people that have a really bad infection that are given it by IV or intramuscular. I was going to say, who's in the hospital right now with sepsis? Intravenous. I mean, if you've got sepsis, you've got bacteria in your blood. It's bacteria. You're given that antibiotic right there into the bloodstream, right where the bacteria are. And then we always have to worry about, are these drugs going to have some type of toxic effect? Again, there are certain things like that antifungal that's going to cause some toxic side effects. Some people have allergies to different antimicrobials. There's quite often people are allergic to a penicillin or a amoxicillin or some type of drug that all the time you'll go to the doctor, are you allergic to any antibiotics? You know already by now. So we want to make sure it's not going to have toxic side effects. We want to know if it's going to have any type of allergic reactions. And we want to know, is it going to have any effect on your normal flora of bacteria? I think this is one of my next slides. Is that as we are taking these different antibacterial drugs, they are killing the infection, but they're also starting to kill some of our good bacteria as well. And bacteria are starting to become resistant to these different drugs that we take. Now, we know that some bacteria are naturally resistant. The beta-lactams, I said, the class of drugs that targets the cell wall, there are some bacteria that have always, to our knowledge, had that beta-lactamase enzyme. And we know that they're always going to break down beta-lactam drugs, and those cell wall drugs are no longer going to be effective. So some bacteria are naturally resistant to certain groups of drugs. They just naturally make certain enzymes against certain classes of drugs. So we can't really do anything about those guys except not prescribe those. What we can start to worry about is the resistance that's starting to form. This is forming somewhat through natural selection and somewhat through genetic recombination. So natural selection, I'm trying to see if this is the picture for natural selection or recombination. Natural selection. So if you're prescribed an antibiotic drug for 10 days, and you got to take it twice a day for 10 days, you've got a sinus infection, you have an ear infection. These are just 2 super common infections that I know people are prescribed antibacterials for. You take it for 10 days, sinus infection. Does it take you 10 days to feel better? Really, you're feeling better like by day 3 or day 4, maybe day 2. that it is not uncommon that people will stop taking their antibacterial by day 3 or 4. They're like, but I feel better. I don't need it anymore. Why do I want to take all 10 days when I'm already better? Well, there's a reason. One is you have a whole bunch of bacteria in your sinuses or in your ear. You have a whole bunch of bacteria. But when you start taking that drug, you start killing those bacteria. The problem is, even though they might be the same bacteria, they're all slightly genetically different. By day 3, you're feeling better. You have killed most of the bacteria, so you're feeling better. But at day 3, have you killed all the bacteria? No. Which ones are the ones that survive? The resistant ones, the ones that are the hardest to kill. 3 days of exposure is not enough to kill them, but then you stop taking the drug. You're like, I feel better, because you did kill most of the bacteria at that point. And what's going to happen is those other bacteria you didn't kill, because you didn't take your full course of prescription, you didn't kill them, so they're going to do what they do best. They're going to grow and divide and reproduce. And now we have more bacteria that are out there that are resistant, that 3 days is not enough to kill these anymore. They're harder to kill. Maybe even 10 days isn't enough to kill them anymore. And this is where we're starting to get some of this resistance. Some are just naturally more resistant than others. And you got to take the full 10 days because you want to kill them all. You don't want to make sure that those slightly resistant bacteria are allowed to live. Now, another way they're developing resistance, so that's just natural selection, that some are just naturally more resistant than others, is that some bacteria are picking up plasmids. Plasmids are little snippets of DNA the bacteria don't need for survival, but yet it's DNA, and that DNA could have information on how to be resistant to the antibiotic. And so they're called resistance plasmids. A little snippet of DNA on how to be resistant. How do bacteria get the plasmids? That's the bacterial conjugation. So the bacteria can attach to each other with a little conjugation pili, and they can exchange that information. I think we got to that point in the video in the first 10 minutes. No, we didn't. Just kidding. We're going to get there. Where we'll see a little diagram where it shows how two bacteria can be right next to each other, and they can share these little plasmids. We'll get to the video. Wednesday or next Monday, we'll finish it. So different ways that bacteria have figured out how to be resistant. Some bacteria now make enzymes that make the different drugs not work, kind of like beta-lactamase is an enzyme against all the beta-lactam drugs. Staph aureus, the bacteria in MRSA, makes different enzymes that inhibit different bacterial drugs. Some slow or prevent the entry of the drug. If the drug can't get into the cell, it can't work. So these things, stop that drug from getting in. Some alter the target of the drug. Some drugs target, say, ergosterol, or they target some other structure, and they will slightly modify the outside so that it can't target that anymore. Some will alter their metabolic chemistry, and this comes down to this picture up here. If we know there's a whole metabolic pathway, the bacteria needs to do all of this to make D, and you gave it a drug that stops that, well, the bacteria is like, whatever, I'll just find another enzyme that can take me from A to C. I'll just skip over B. I don't care. It will alter their metabolic chemistry so that it skips over all of that and it will still make their end product. Cool for them. Drugs out, that as the drug gets in, they truly have pump mechanisms just to pump the drug right back out. And some bacteria can make biofilms. That's the stuff on your teeth that you guys saw on the scrapes. That, yes, they stick to each other, they stick to surfaces, and they prevent those drugs from even getting anywhere near the cells. So what's happening is that we are starting to get, I've got lots of pictures on the slide that come up. We're starting to get what's known as bacteria that are resistant to lots of different classes of drugs. And so we'll call them superbugs, sometimes multi-drug resistance pathogens. A superbug just means a bacteria that's resistant to at least one class of drugs, meaning this is a bacteria that's resistant to all the beta-lactam drugs, which means you can no longer kill the bacteria by giving it one of those beta-lactams that inhibits the cell walls. Usually it's because they pick up a resistance plasmid that targets that one specific mechanism of action. The top places that have superbugs, these resistant microorganisms, are hospitals and nursing homes. And I guess why? We use the most antibiotics, and this is where all these sick people are. So you have lots of people in a very small area. They're all sick, and they're all getting prescribed antibiotics. I mean, it's like the perfect combination of, let's have all these bacteria. They can all talk to each other because they're so close to each other, and we're all given them different kinds of antibiotics, and it's the perfect breeding ground for resistance to happen. And so we're creating superbugs in these environments, and this is where we're going to have the highest number of superbugs in hospitals and nursing homes. The different kind of places that you guys might be working. Something called a multi-drug resistant pathogen just means it's resistant to at least 2 classes of antimicrobial drugs. Or maybe it's resistant to all the drugs that inhibit cell walls, all the drugs that inhibit protein synthesis, the 2 types of classes of drugs. And then we have something called cross-resistance or pan-resistance. That's what they called it in that movie we got to, that part. It means these are drugs that are resistant to all antimicrobials. They're resistant to everything across the board. And those are scary because then we truly have nothing to treat it. We just hope the immune system can get rid of it, and that's all we have to work on. Okay, there's so many different superbug little comics. So things that we can do to slow the resistance from happening. One is to maintain a high concentration of antimicrobial, for long enough to kill everything. It was that picture of, make sure you take a high enough level that can kill the bacteria, and you take it for the full course that it's prescribed, the full 10 days. Because you want to make sure you kill them all. Even if it kills normal flora, sometimes you do actually have to kill normal flora to make sure you kill all the bacteria. Sometimes this also requires taking antimicrobial agents in combination. You're not prescribed just one antibiotic, you're prescribed 2 or more antibiotics or 2 or more antimicrobials. And the idea is they all work together to kill a certain type of microbe. That's usually for some of the hard to kill ones. If they're already hard to kill, we want to make sure we throw everything at them to kill them, because the longer they stay alive, the more chances are they figure out how to be even more resistant. So it's, maintain a high concentration, throw everything at them, and then the reverse is also just not using antimicrobials if you don't have to. So if there is a bacterial infection, kill them. But if there isn't a bacterial infection, don't give them or expose them to antimicrobial drugs. Every time you expose any type of bacterial to antimicrobial drugs, you are giving it the opportunity to figure out how to be resistant to it. So only use them when absolutely necessary. It used to be, I'm old enough to know that, yes, it did used to be that if you went into the doctor, you had the flu, they're like, oh, you're sick, here's some antibiotics. Antibiotics don't work against the flu. Bacteria, virus, 2 totally different things. But people felt better when they left the doctor with a prescription, even if it did absolutely nothing, and we realized that we were increasing that resistance. We were exposing normal flora bacteria to antibiotics, that were developing unique resistance methods that were sharing it with pathogenic bacteria. So now we have to come up with new variations of drugs that we already have. We're having to try to find new antibiotics, which is pretty rare to find new things out in nature. We're mostly taking a lot of these things that we already have and modifying them to try to make them, effective against what we have. I think that's not a slide. More doom and gloom. I'm going to hand you, she missed, but I'll email it to you. Online quiz for this unit. There's just this. So there's not even like a big test or anything. It's just a worksheet. So it's using the chapter 11, 12 notes, not chapter 9, 10. Use the PowerPoint, use the notes, use your e-textbook, and answer the questions, and bring this back on Wednesday. And I think that's all you have to do for Wednesday. It's just this. No online quiz or anything else. you guys are not even a couple minutes in. Wasn't sure if I was going to get through it or not. Because of that, there's a ton of resistance. So I want to say, yes, penicillin is still prescribed, but it's still been slightly modified. I mean, they don't consider it a semi-synthetic because it's still pretty closely related, but yeah, it's still prescribed. Our biggest issue, I don't know the percentage, but a good number, I want to say 15% of the population about is probably allergic to penicillin. Oh, it's not fun. Yeah, but I know quite a few people, because usually when I ask, I usually in my classes will have one person, you guys are a small group, that's allergic to penicillin or amoxicillin, or even, usually if you're allergic to one, you're slightly allergic to another one. They get like rashes and hives and stuff like that, but that's why a lot of times it's not prescribed, is mostly because they know. If you don't know if you're allergic, you don't want to find out that you're allergic. And there's other things that we currently have. It's beautiful today.